Okay, so I'm just actually introducing this second part of the afternoon, not talking directly about CTD, but introducing, I think, some of the questions that we all have, and based on other things, other disorders on which I worked, and, well, successes and, I guess, obstacles that we faced. And again, this is really food for discussion. So when we talk about those challenges, I guess the first thing that we are asking is, what do we measure? Are we talking about a therapy which is going to have a symptomatic effect? So for example, I'm going to talk about GLUT1 deficiency syndrome, so a deficiency of glucose import into the brain, where you have a lot of episodes, including movement disorders, for example. When you correct the energy, you see a reduction of those movements, so it's very symptomatic. But then, and especially here, I believe we are talking more about disease-modifying therapy, for which the effect on symptoms is very much unknown, and there is a matter of time of how long we need to observe these changes. But I guess this is the first question that we should address. The other one is which tools we are using, and we know that regulatory agencies want to see mostly clinical readouts. So when we come with great ideas with even surrogate biomarkers, which mean biomarkers that we believe really follow changes in clinics, the agencies are not really open to these ideas, and you may have the best molecule and the greatest design. If in the end the agencies don't want to hear about it, you have done all of this for nothing. So it's important to also keep what they have in mind. But the idea is that biomarkers may be a first step towards something else. Then the question after, which is more of a, I won't go into that, statistical question, how many patients do we need to see an effect? Because if we are just about to see it, but we are lacking some patients, then all the efforts that were done at our risk also. So it's based on the effect size. If you have a marker that changes a lot, then you need quite less patients, and I will show you that. Then there is the question of the design that we want to use. Of course, we are talking mostly about trials where we have at least a placebo and something else. I mean, a molecule which can have one or sometimes two doses or else. But in the field of rare disease, there are a lot of possible designs, and there are, as Rima Nabou this morning alluded about, there are actually a lot of research projects just on this. And then what is the cohort that we want to target? And here again, we can decide what we call to stratify. So if we think that we have two different populations and we cannot mix the observation from these two populations, we can predefine that. But then, of course, you lose power in the statistics in the end. And a question we may have, and we had a dedicated session for girls with CTD this morning, is whether or not we are talking about boys and girls trials at once, or how do we do that? Do we need to, or can we stratify? So about the question of effect size, this is, I worked a lot, I'll tell you a little story about this on Huntington disease, which is a rare disease, one of the model of neurodegenerative disorders. It's 100% genetics. You have patients that are pre-symptomatic, so they know, as we can know in CTD, what are their gene status, but they have not yet developed a disease, unlike in Parkinson or Alzheimer's, where we usually don't have that. Even there are rare cases that are genetics as well. And so in Huntington, there has been a lot of research because it's more common than CTD, to actually design trials. There is already gene therapy that have been started, and there are a lot of trials ongoing. And so they showed basically very nicely, this is what you use, one of these markers, whatever it's clinical or biomarker. And let's say you have an effect size, so basically how much your marker is going to change. Here around 0.6, you are going to need 400 patients per group. So let's see if we define that, or the effect size of whatever, or a PET study, or the infrared spectroscopy study is of 0.6 or 0.7, we need that many patients. So obviously we can't have that, providing that we want to see a 40% effect. Of course, if you want to see less of an effect, you need less, but still a lot of patients. But then if you have a marker which is very sensitive to change, like let's say 1.5, then you only need 50 patients per group. And then it becomes realistic, at least for some of these diseases. This is something important to keep in mind, and that's why you really need to know in your population how your marker, whatever it's clinical or else, imaging or biological, behaves, because you have to determine this effect size problem. So to go back to some treatment development, I've been involved for 15 years. You use this compound called heptanoate, which is basically some fatty acids. You may know that for fatty acids, we have what we call long chain and medium chain and actually short chain fatty acids. Usually what we eat is mostly of long chain, but there are some also medium chain that we can find. But this one is actually not from the food, it's synthetic, because it has an odd number of carbons, so it's called heptanoate because it has 7, actually, carbon. And we've been interested in working on this, with this compound, in this disease I mentioned, GLUT1 deficiency, where the import of glucose, we talked before about PET for glucose, who actually uses this transporter. So when glucose doesn't enter properly into the cells, and this is a dominant disease, so you have a version of the gene which is functional and one that is not, because if you have no transporter at all, then of course it's probably not viable. And so patients have a lot of episodes related to this brain energy problem. So it's a good model to study energy deficiency. And when we use this compound, which comes in the form of heptanoates, it's called triheptanoin, we actually bypass completely the problem of the hypometabolism of glucose because we rely on this medium chain triglyceride. And actually, it doesn't even need a transporter. It can go through passive diffusion into the cells. And it provides substrates as other maybe therapies that you know, ketogenic diets, the ketone bodies use other transporters to get into the brain than GLUT1. And here I was telling you, you may expect a symptomatic effect. And this is what we showed in 3 consecutive studies. And we have more and more data that we wish to submit to the French Drug Agency to, at some point, get, we hope, some approval for that, is that you have a dramatic response, more than 90% reduction of the movement episodes that this patient have. So it's basically torsion of some limbs or impossibility to speak or falls or gait problems. So this is easy also to measure. So then we wanted to use it for Huntington, but without going into the mechanism of this disease, the protein itself, the huntingtin, actually is involved in all the process of neurodevelopment before it's even a neurodegenerative disease. It helps cells to make their division. It's kind of a scaffolding proteins which support, like for a building, all aspects of cell metabolism. So it has so many functions, and one of the consequences is energy deficiency. So we were not hoping with this treatment, because let's say we can use maybe also this drug for the energy problem in Huntington, we were not hoping to have a symptomatic effect because there are so many other things going on. It's not like in GLUT1 again. So it's one aspect of the disease that we wanted to address. And in Huntington disease, actually, there were, as I mentioned, a lot of therapeutic development, gene therapy, but many other strategies. So at some point, you even become, and actually we had this discussion within the association, that CTD may be a disease where at some point there will be not enough patients for the number of trials we want to do. And you may think, well, that's this. When we get there, we are lucky. You're right. But it's also makes sometimes things complicated because you still need enough patients to see something. So in Huntington, this was getting a bit of the issue. So we really needed to be sharp in what to do. And we were not going to do a 1 year or 2 years trial based on our hypothesis. And of course, there were much more data we had to say that maybe heptanoate could be a good therapy for this disease. So we used actually phosphorus spectroscopy that Pierre-Gilles Henry introduced to you, so not the proton, but the phosphorus, because we were interested in looking at the ratio between phosphocreatine and inorganic phosphate, because this ratio reflects changes in energy dynamics. And what we did was to measure the metabolism of basically the brain in the occipital cortex during a very simple task, just some flashing checkboard that patients had to see, and which activates that region, and see what are the changes, first of all. And so the first thing we showed is that in Huntington, you don't see a normal profile like you have in the healthy population. So here you see 3 different populations. The first one in controls, it's at rest. Then you activate this occipital cortex, and you see an increase, which is only 10%. Yeah? It's not huge, but you see, and we reproduce that when the brain is activated in that area of vision. And then at rest, it goes back to baseline. And in patients, we did not see that. So it was actually the first real demonstration that there was an energy problem in this disease. There were a lot of data in animal models and stuff, but nothing, data on patients who died, but nothing real in real life. So that was, for us, a big thing. We could reproduce that. And then we just did a 1-month trial using our drug to see the effect. And we saw a recovery of the normal energy profile. So that doesn't mean that it is going to long-term do something, but at least the hypothesis was correct, and we could see that with our drug we could correct that. And so we just completed, and with COVID and everything else took more delays, but we did a study in 2 centers in the Netherlands and France with a total of 100 patients. And over 1 year, we see that not only we can stabilize the motor dysfunction, so Huntington is also known as a chorea, so large movements of the limbs, and this is captured by a score, which is called the UHDRS, which stabilize. Usually it increases with time, but there is a region in the brain that is very known with Huntington to get atrophy with time, and this is the caudate, and we saw a 50% reduction of that atrophy over time. And by the way, in the meantime, the first gene therapy trial, which we think, okay, with gene therapy, we're getting there, right? Completely failed because actually it's even aggravated patients because it was not just targeting the mutated protein, but the whole protein. And now we know that Huntington has also an important function, so just inhibiting that is not the solution. So I'm saying that because people were kind of smiling, even maybe laughing a bit at the ideas that, well, some maybe fat can use this patient, but at the moment there are not so many successful trials so far in this disease, and we are one of them. But it took, many years of development to get there. I'm going to talk briefly now about another example, which is actually a disease that was mentioned this morning, about this adrenoleukodystrophy, which is another X-linked problem. Here we are back to actually some fatty acids. It's the transporter, which is mutated, is using the long-chain fatty acids to get into a place in the cell, which is called the peroxisome. And from there, those fatty acids go in the mitochondria to produce energy. And in this disease affects all cells of the brain, but we mostly think about neurons when we think about a neurological disease. But you know, because of creatine, actually metabolism, that other metabolic cells are very important. But we forget about a component which is actually the immune cells of the brain, which is called the microglia. And it happens that because of the mutation of this protein, the immune cells of the brain, as the immune cells actually that we can also find in the blood, which is called the macrophage, they are stuck into a pro-inflammatory status. So immune cells have to be inflammatory at some point and then regenerative. But this switch is controlled by metabolism. And when you have a mutation in some protein that actually affects metabolism, you can have your cells bias into this inflammatory status. And the company we work with developed a drug that is not only going to help the mitochondrial dysfunction of this disease because of all the problem of fatty acids, but also have an anti-inflammatory effect. So that looked very promising, and the drug developed by Menorex is also actually reaching the brain, which is a big problem in this disease. And so for that, in this disease, it was mentioned this morning, the life-threatening condition that only affect men is this very huge inflammation that leads to death usually in 2 or 3 years. So you monitor that with MRI. But with standard MRI, you see when this is just starting and this is over some months, you see the white part here. This is the beginning of some inflammation in the brain of that 21-year-old patient, but you see not much. So we used the technique that was mentioned just before about this diffusion tensor imaging, which use propagation of water to analyze the content of fibers in the brain. And we are working with another biotech that actually can do that on just one single patient. So you can have a map of the fiber trophicity, of the number of fibers in the brain just for one patient, and you can follow that over time. And this is the changes we see, actually, unlike what we have with our eyes. And again, you don't need controls patients, you just need this one person because they have standardized, calibrated this tool. So this is what is powerful. And you see that after 3 months, it's actually aggravated. 3 months after starting treatment, but then it started to slow down. And actually, we went back after 1 year to the images that we had at the beginning. So we have fully stabilized this inflammation demyelination. About the tools, clinical or biomarkers, it was also mentioned, and I think this is really important. The regulatory agencies want to hear about patients' opinion, and you will say, well, that seems logical, but it has been forgotten for decades in research what actually patients feel, right? It was what the physicians feel. But the quality of life scales that mostly have been developed have been actually designed by physicians. So it's complicated, right? Because how can you take patients' perspective into account? But one thing that we got to realize when we work with psychologists who are doing research in neuromuscular disorders, they were having new treatments, new approaches that were actually working, improving some of the muscle function in some of these genetic muscle disorders. But the patients on the quality of life didn't show anything. So I was like, why is that? Because your grade of happiness or whatever, fulfillment in life is this difference between what you experience and what you expect. And that's why you can have very disabled people who have actually great quality of life because of this adjustment, and at the same time, people who seem to have everything and constantly unhappy. But if you enter a trial, even if your experience improve, but your expectation actually also has moved away, the gap is the same. So you don't perceive that you have benefited. And that is a lot of the issue around those quality of life that the agencies want to see, but that a lot of them fail. And that's why came this development of qualitative research, which is called, this is just one example, in particular one that I really like, that the people working in rehab for a lot of patients with neurological disorders use, which is the goal attainment scale. So the idea is that patients or caregivers, when patients cannot do that, identify, for example, 3 meaningful daily activities with performance problems, and you define before, so it's different for each individual patient, what would be, and you rate an improvement, a small or a big improvement, or at the opposite, something that will deteriorate. And in the end, this is a way to capture in real life what can be meaningful for a patient. And sometimes when you have a drug you want to test, it's just one patient. I saw just one of them actually 2 days ago. We defined before what will be meaningful for her. And for example, she could not pick anymore any pack of water because of muscle strength issue. And that was a major issue. The second thing was the length she could walk to go shopping. And we defined before what would be a little improvement or a big improvement. And this is, since this is predefined, you cannot say after that, oh yeah, it's because you want to fit that into your expectations. It's what we have agreed on. But it's for her, it's not for somebody else. Just because we are going to talk about natural history and things like that that provide actually some of the material to design studies, what we don't want to have is to miss the primary endpoint, because then the agencies don't like it, even if you have great secondary outcomes. So going back to this compound, it was actually tested initially to improve the gait in those patients with the myelopathy form of the disease on the 6-minute walk test. Why? Because there were only data available on that for many years. So when they had to define how many patients, they used this scale. But it failed. Instead, it improved the proprioception of the patients, which is related to the myelopathy, using those tests with eyes closed and feet apart, etc. But the striking thing, going back to this very severe demyelinating inflammatory disease, is that almost all of the patients who converted to this cerebral form were on placebo. So for us, it's a major thing. Still today, 3 years after all this data, agencies won't hear it because the primary failed, because we didn't expect that. We didn't expect to see that. So now we need to do a trial in the US where the outcome is survival. So patients on placebo, knowing that this disease can kill them, because the agencies wants to see, and it's survival, the outcome. I mean, we've been in discussion with them, it's just terrible. This is just to tell you the impact of how we should prepare these thoughts before. So to finish with CTD trials, of course, I believe that what we are talking here is much more of a disease-modifying therapy than a symptomatics. So it doesn't mean that we cannot measure effect on symptoms like cognition, like behaviors, like else, but the effect size is probably going to be small, right? So it is, of course, an issue. We are, especially at the beginning, we don't want to exclude, let's go for clinical because that's what the agency wants, or let's just only do biomarkers because we definitely need the two. How many patients, we don't know. It will depend on what will still be used to be the tool to determine actually the sample size, the design even less. Which patients, again, we had the discussion, boys, girls, both, if we stratify. But my messages would be that we need natural history data, and Aurore will be presenting that. But one thing to keep in mind is that sometimes in natural history data, you find a marker that seems to change quite a lot or can be informative. But yet, maybe your treatment is not going to have an effect on this. And so that's why you need to also try to get preliminary data to see, okay, I know that I'm trying to compensate for this metabolic problem, but still, what does it do? We have to have some kind of pilot information to know where to go, because we may have a marker from natural history that say that looks like the best one, but actually the drug is not going to help on this. And think about the Minorex trial who failed because of that. I really promote these goal attainment scales because I really believe that, not as a primary, of course, endpoint, but I think that they are very useful in addition to everything else we can, and they are very simple to establish. I also am an advocate of metabolic imaging to have this first idea of what we do when we target energy metabolism like here. So I showed you an example that we had in Huntington or other tools that we are using for diffusion. Ideally, we would want to go to pre-symptomatic, right? If we can, we know that for GAMT, now there is this, of course, newborn screening. In the years to come, we may have that for CTD if we have a treatment. The thing is that for now it's too early because the agencies first and always want to see data in symptomatic people before you go to pre-symptomatic. And we learned that in Huntington, and that was very sad because we were really hoping that we would be able to get some waiver from that, but they won't hear it. So my last comment would be that I really think we should need, and this is just an opinion to be discussed, but a pilot study with any drug that will come for CTD to first get a sense of what it can do on on imaging, biomarkers, whatever the ones we've discussed before, and clinical. Something short, not many patients, but just to have a first idea of what this drug can do before we get into the big thing, and so that we don't miss the big picture. Thank you.